Hello, everyone, and happy holidays to all. Welcome to the Phytobiomes Alliance webinar series. My name is Dusty Gallagher, Executive Director of the Phytobiomes Alliance. We have an excellent webinar for you today. And to begin, I would like to share with you a little bit about the Phytobiomes Alliance. The International Alliance for Phytobiomes Research is a nonprofit consortium of industry, academic, and governmental scientists. And our focus is on the complete systems of plant-based agriculture, which we call the phytobiomes. And the phytobiome consists of plant agriculture in a particular substrate. And for this example, we're going to use soil, but it could be many other types. And then we focus on all of the components that are linked to the phytobiome. We have animals and plants, microbiomes, macroorganisms and macrofauna. The climate, weather, and water available is also a significant linkage to the phytobiomes. And then all of this is influenced by management practices. And so what we do at the phytobiomes is we identify the major research gaps between all of those linkages of the phytobiomes, and we try to address those gaps with systems-based research. Examples of the phytobiomes include traditional crop-based agriculture, but it could also include animal agriculture, pastures, containerized agriculture, forestry, and vertical farming. And so our vision at the Alliance is that by 2050, all farmers have the ability to use predictive and prescriptive analytics and information based on geophysical and biological conditions so that they can determine the best combination of plants, management practices, and inputs for a specific site in a given year. And we do this because of the generous sponsors that we have with the Alliance that you see here on this graph. Without them, we couldn't do what we do. And they are very supportive of this webinar series that we have. And so we want to thank them. And we have a couple of upcoming events that we would like to share with you. In early 2025 at the Plant and Animal Genome Conference, we will be holding 2 workshops. One on Sunday, January 12th, exploring phytobiomes, and on Tuesday, January 14th, the uniqueness and commonalities between plant, animal, and soil microbiomes. So if you are attending PAG this year, we hope that you will join us at our workshops. And our next webinar will be in February of 2025. And so please stay tuned for more information and registration information. It will be available soon. And so here's a few housekeeping items for our webinar today. This webinar will be recorded and will be posted on the Phytobiomes Alliance YouTube channels later today. And following the presentation, we will have a Q&A. And so we would like to ask you to submit your questions in the Q&A panel, which is different from the chat panel. Please use the chat panel for only messages, to see messages from the organizers, and if you have any technical difficulties. And the questions panel is the most important, and that's where your questions will be asked to our speaker. There's also a handouts feature, so there will be PDFs of the slides presented today, and any links that will be presented in the slides will also be added to the chat panel. If you are not already registered to our mailing lists, there is a URL for you to do so. And this is where all information about the Alliance will be shared, including information for our February 2025 webinar. So I just want to take a moment to thank you for your participation today. You can also follow us on X or Twitter and LinkedIn. So that is another area in which you can find information about the Alliance. So without further ado, I would like to introduce our speaker today. And we're very fortunate to have Kirsten Hofmockel, who is with the Pacific Northwest National Laboratory here in the U.S. She's the Chief Scientist for Biological Sciences Division, Earth and Biological Sciences Directorate. And she will be discussing how we decode the rhizosphere ecology for soil health. So I welcome Kirsten to the screen. And I will stop sharing my slides. And Kirsten, it is all you now.
Thank you, Dusty. And thanks everybody for joining us today. And thanks to the Phytobiome Committee for this invitation to speak. It's such a great seminar series I've been following. And so today I'm going to share with you research that we're doing to try to understand how rhizosphere ecology can promote many of these beneficial functions that Dusty was talking about in her introduction. And they're really the goal of the Phytobiome Alliance. So as anybody who's like a phytobiome fan knows, the rhizosphere is a hotspot for biogeochemical reactions that are important for plant productivity, nutrient cycling, and soil organic matter formation. And many of these functions, these phytobiome functions, have impacts that scale beyond the plant microbe interactions to influence larger ecosystem outcomes like carbon sequestration, greenhouse gas emissions, and even potentially the bioeconomy. So our research is highly collaborative at the national labs. So everything I'm going to present to you today has been developed by a crew of folks at the National Lab, at Pacific Northwest National Lab, and some of our external collaborators at Washington State University and the National User Facilities. The work has been funded by the Department of Energy, and specifically in the Biological System Sciences Division of the Department of Energy. So research goals are basically, in our group, are to link microbial interactions to ecosystem outcomes. So we want to discover these molecular reactions that regulate ecosystem functions and enable the prediction of some of their probable responses and even feedbacks to environmental systems and environmental change. And so much of our work happens at this molecular level, where we're trying to understand plant microbe interactions and how that might be affected by, as well as affect, microscopic interactions. So this is sort of, microbial communities and those interactions that are happening right at the interface, how that influences and is influenced by the core to pore scale, and finally, how findings from our field sites or our experiments might relate to other soils, other ecosystems at the landscape or larger scales. And I just want to take a moment to familiarize you with the national labs in case this is the first time you're hearing about them. I work at Pacific Northwest National Lab. We have lots of university collaborators, industry collaborators. It's a very welcoming community. All of our work is team science, so we're used to interacting a lot with different kinds of people. There's lots of internships for undergraduate researchers, and as well as like postbacks that can come and work at our facilities. There are graduate student research fellows through the Department of Energy, and you can go there and find these sites or reach out to me if you have questions. We do a lot of mentoring of postback, postmasters, and postdocs that come and work at the lab as our staff, and that's our way of sort of developing the workforce. There's ways for professors and industry partners to collaborate on organizing sessions at society meetings or collaborating on research for funded programs. We can do subcontracts either direction if there's work that we want to do together. We can also support things like sabbaticals for faculty or joint appointment. Just a quick outline then of what I'm going to present today. So the work is from our soil microbiome science focus area. And this is a large long-term research program, and we're working on many facets of rhizosphere ecology and how we might develop this predictive understanding that could inform how we manage ecosystems. So I'm going to share with you a little bit my perspectives on rhizosphere ecology. I'm sure most of you are familiar with it, but sort of the angle that I'm coming from for this talk and our research. I'll spend some time on coupling field and lab experiments That's a big theme in our research as we try to move across scales. So in order to have this molecular understanding, a lot of that might be lab-based. How does that scale to understanding things in the field? How do phenotypes that we identify in the lab translate to the field? And how can we decode the phenotypes we see in the lab to understand the molecular or genomic basis of those? I'm going to share some new unpublished work on rhizosphere metabolipidomics that we're really excited to get people's perspectives on, and then a little bit on future research and opportunities. So there's been a major science investment in managing rhizosphere ecology for beneficial functions, like increasing crop yield while reducing greenhouse gas emissions and building soil organic matter. And our group is focused on discovering that fundamental biology that, one, mediates beneficial rhizosphere functions, and two, promotes resilient ecosystems to environmental perturbation. So this is a figure from one of our 2020 papers where we outlined some key areas where plant microbe interactions can influence beneficial functions. So things like generating extrapolymeric substrates that can help with water retention, that can help with resiliency when there's drought or limited water resources. Microbes can be a sink for rhizodeposition. And as those microbes grow and turn over, some of those residuals can persist in the soil as microbial necromass and even potentially contribute to carbon sequestration. And then microbes can play an important role with plant growth promotion with molecules like indole-3 acetic acid or other plant growth promoting hormones, or through symbiotic relationships or asymbiotic relationships with nitrogen fixers. And these molecular interactions have outcomes that are important for short and long-term responses to climate change. So as depicted here, in the short term, as elevated CO2 and temperature might cause an increase in plant productivity, which will result in an increase in rhizodeposition, mineralization, and potentially respiration. And in this short term, we expect there to be more carbon stored in the soil because any of that respiration would be offset by increased plant productivity. But over the longer term, as that easily available carbon gets depleted, the microbes might mineralize more of the carbon reserves. And that will result in a feedback with greenhouse gas emissions. And this is particularly a problem in boreal wetlands, for example, or thawing permafrost that contain large reservoirs of soil organic carbon that are becoming increasingly susceptible to microbial degradation as the world warms. In more temperate latitudes, a central question in phytobiome research is how we can draw down atmospheric CO2 and potentially sequester carbon while maintaining plant productivity, and do all of this in the face of a changing climate. And we're especially interested in drought stress. So in many regions of the world, as you can see here, there's increased incidence of extended drought, and this creates considerable pressure on global agricultural yields. And while many plant breeding systems have selected for drought resilience, there is a whole world below ground that is yet to be incorporated into our plant breeding programs and our management systems. And so the work I'm going to share with you today is from Eastern Washington, where plants frequently experience drought stress. And our research is supported by Washington State University in the Irrigated Agricultural Research and Extension Center in collaboration with Dr. Steve Norberg. And we use this irrigated field trial in Prosser, Washington to test hypotheses about drought effects on rhizosphere ecology. So our site focuses on tall wheatgrass. This is a perennial bunch grass. It's a deeply rooted plant, and it's well adapted to drought stress. And it grows really well on our marginal soils here. So we're working in aridosols. The pH is about 8, and it's only about 1% soil organic carbon. So we're interested in how we might be able to sustain plant productivity and increase carbon storage below ground. One of our key questions then is how water availability or water stress can change rhizosphere interactions. So our experimental design has a bare plots with no plants, so we can detect the plant effect. We have bare soil that's irrigated, so we can detect what's the water treatment and separate that from the plant. And then we have 2 irrigated treatments, one at 25% of field water capacity and the other at 100%. So our low and high irrigation treatments. And anyone with a passion for soils knows that variability is the signature for soil ecology. And a major knowledge gap in this variability for rhizosphere ecology is the below ground phenology of perennial rhizosphere ecology. To address this challenge, we conducted a monthly sampling campaign from June of 2023 through July of 2024. And most of the data I'll present to you today focuses on the top 15 centimeters of soil. So you can see here the soil temperature. If I translate for US, it ranges from about 40 to 77 degrees Fahrenheit. So in the summer, it's not uncommon for us to have triple digits for multiple weeks in the above ground, like in the air temperatures. We have 2 irrigation treatments, like I mentioned, 100% and 25% of field water capacity. And so this irrigation treatment causes a significant difference in our gravimetric water content over the year. We only irrigate during the growing season, which here goes until about late October. And then we turn off irrigation, and that's kind of when we enter our wet season. So you can see that the gravimetric water content converges. And then when we turn irrigation back on in the spring, they start to separate again. The irrigation has a strong influence on pH. Remember, these are iridosols. These are high pH soils. And so we see a lot of leaching of cations with the high water treatment that's reducing the pH. And finally, we see the root mass varies over the field season. And one important part about this potential bioenergy crop is that it's harvested twice a year. So we have one harvest that's at the end of July and another harvest that's at the end of October. And so it's interesting to see how that above ground harvest is reflected in what's happening below ground. And here we see a real difference in the water treatments on below ground root production. And so we want to understand how those changes might influence larger scale outcomes like respiration or carbon sequestration, and that balance between loss and gain of carbon. So as a first step, we did in situ soil respiration measurements. And these were done by Sharon Zhao and Kaylee Cornwall, who were interns at the lab. And you can see here the respiration measurements in this top panel. We see a strong seasonal response that also reflects this harvest. And that this is reflected in the root mass. And so we see these similar patterns in the root mass from root picking and the CO2 emissions. Interestingly, we do see a significant water treatment effect on the root mass, but we don't see that in the CO2. And we know that there's another component to that respiration, which could be the microbial contribution. So we find that these are intriguing results. Even when root biomass is low, the respiration rates seem to be maintained. And so imagine if we could manage this carbon loss through rhizosphere respiration. So instead of carbon that was shunted into pathways that could build soil organic matter or even sequester carbon instead of being lost to the atmosphere. So in order to basically understand those plant microbe interactions and what might be driving respiration, we looked at substrate-induced respiration, and it reflects the potential microbial contributions to soil respiration. So we looked at a panel of 31 potential substrates that are representative of soil metabolites based on literature and ongoing experiments from our group. And this is led by Cheryl Bell, who leads our field research, and Evan Warburton, who is a postdoc intern. And Sharon and Evan created this custom substrate-induced respiration plate that includes these main classes of molecules. And when we inoculate the plate with soil at each monthly sampling date, then we can measure the respiration induced by each of these substrates. And what we hypothesized was that the response would reflect what microbes in the field are primed to metabolize based on what they're exposed to at that moment in the field. And so, for example, we might expect highly bioavailable compounds like amines or amino acids, even signaling molecules. Those would be rapidly and consistently metabolized, especially during the growing season, while more complex substrates might be more important when roots are turning over and those substrates are important. And so what we found here was that microbial respiration reflects this potential, the respiration potential reflects substrate complexity. So here you can see on the left, we have these easily metabolizable compounds. We found that glucose was pretty consistently available. The microbes were ready to respire that throughout the year. Oxalic acid was largely available. You know, there was a downturn here in sort of the winter months, but during the growing season, that was highly available. Well, as things like chitin and this olate, which is a wax, we find are available mostly in this winter and fall season when maybe less bioavailable carbon is there from root exudates. And so in the field, we know there's this combination of rhizodeposition and root turnover, microbial necromass, that creates this complex cocktail of substrates that microbes are exposed to. So to better understand this seasonal variability in a more integrated fashion, we combine the 31 substrates over time, and we plotted it here on this NMDS, where each dot represents the full respiration profile of all 31 substrates for one soil sample from one field plot and one date. And the colors then are for each month that the samples were collected. So for each date, there are 6 triangles for the 25% moisture treatment and 6 circles for the 100% irrigation treatment. And we see that there's very strong seasonal signals. So if we start here, January and February are sort of in these like purple area, March through June are in these like pink and yellow and blue and green. Then we find July through October, which is in the peak growing season, are these warm colors up here. And November and December are these light blue and light green colors. So there's definitely a strong seasonality. And we found that representative exudate compounds explain the seasonal variation in microbial respiration. So here we can look at the loadings of substrates that are significantly structuring the respiration patterns. Each vector here represents a substrate, and it's scaled by its correlation to the respiration profile. So here on this graph, we have the top 10 of the 31 substrates that had the strongest correlations and are represented with vectors. And what we find is that organic acids differentiate along NMDS1, and that these plant polymers or microbial polymers here differentiate along NMDS2. So this gives us insight into how substrates, microbes might be metabolizing substrates, and there might be a strong seasonal signal to this. So if we circle back to the individual compounds then, looking at some of these signals that were, these substrates that were highly influential on the distribution of the respiration patterns, we see here that the seasonality in these, in the respiration of these substrates, it aligns with the phenology of plant roots that are here in this lower right panel. So there's 2 significant growth periods, right? Because we have a harvest, 2 harvests that happens, and we have 2 significant growth periods of root activity. The solid is the above ground, and the dashed line is the below ground. And so we see that this really corresponds to the peaks in this substrate-induced respiration. And even though these are potential respiration rates, they still demonstrate the importance of the plant life cycle when managing CO2 emissions. And especially in the winter months, when we might, when there is a potential for respiration. And this could be quite high and quite impactful, finding when we consider how climate change might be warming the world. So in the previous study, we focused on model rhizosphere substrates based on our studies and the literature. But there's still so much to be learned about the rhizosphere. And Snehikovian here is leading our research on the molecular characterization of rhizodeposition in perennial plants. And one of the grand challenges in decoding root metabolism is that research tends to be performed in hydroponic systems. Typically, these are with annual plants. These are model plants like Arabidopsis or Brachypodium, and they're only for a few months. They're only a few months old. And those data are very important, and they help us to understand plant metabolism and build databases. But we also expect that there might be strong differences in rhizodeposition with plant age, and especially when we consider perennial plants, where seedlings are really quite different than the well-established system. So here, for the first time, we characterize the exudate profile of mature, field-grown perennial plants. And so our plants are 5 years old, and they're well-established in the field. And what we did is we collected them from the field, gently excavated them, and then separated the bulk soil from the root-associated soil. Then we washed the roots and got a root wash soil. And then after we washed it, we captured the exudates in hydroponics. So what we're doing in this experiment is trying to understand the gradient of metabolites and microbial interactions as they move away from the rhizosphere. And what we find is the metabolipidomics provide us really important molecular resolution for understanding what pathways may be contributing to different outcomes like respiration or soil organic matter formation. And so here we have two approaches. One is using this gradient that I just explained. We're looking at the bare soil from the field and comparing it to the bulk soil, root-associated soil, root wash soil, and then the root exudates. And we're looking at them for both metabolomics and lipidomics. And aside from the technical challenges of sampling root exudates, a second major frontier is that we are unable to annotate many of the compounds that we detect. So this is a really emerging field, especially in the environmental sense. So most of the databases are built around human health, and there's no central database for plant microbial or soil metabolites. And so it's a lot like where the field of metagenomics was like a decade or two ago. And so for this work, Sneha mentored two Suli interns. And what they've graphed here is the distribution of metabolites in the different superclasses for exudates in soil. And what's important to know is that we are really taking on this challenge of the unknown molecules. And so what we did was we used two methods to classify our molecules. First, we used a standard MSMS spectral library search. And you can see the unique number of compounds here. We're 226 for the exudates and 218 for the soil. But in addition, we used a deep learning approach. So this is an artificial intelligence approach that we can class assignment into, and this has substantially increased the characterization of the chemodiversity of both exudates and soil. And so you can see here, the soil went from 218 to 6,813 compounds that we were able to identify. And the exudates went from 226 to 1,205. So we're really excited about these results because this is probably one of the most comprehensive annotations of rhizosphere metabolomes to date, and especially in a mature field-grown perennial plant. So in addition to the metabolites, our crew is becoming obsessed with lipids here. And so we're discovering there are diverse lipids in root exudates. This work is in review right now, and we have a bioarchive article posted. Lipids are basically hydrophobic classes of metabolites, and they tend to be quite sensitive to changes in physiology as well as in response to stress. So most of these lipid classes have been measured in microbes, but this is the first to characterize intact lipids in root exudates. And we discovered the presence of a diverse variety of lipids, and including a substantial level of these triacylglycerols. And if you're interested to learn more about this, you can check out our preprint. And then when we compare this lipidome of the root exudates to the root itself, here we have the same classes of molecules in the root. And what we see is that there are about 91 molecules that are unique to the exudates, and about 327 that are unique in the root, and 176 that are shared in both the roots and the exudates. So it's not clear why certain molecules were detected in the exudates but not observed in the plants, but this has been shown in other model plants. And we're excited to learn more about how this shift from the root into exudates, how those unique molecules might select for microbiomes and different metabolic pathways. So one of the themes that we're really interested in is how do those outcomes scale to influence larger environmental factors like the soil structure, the soil carbon storage, greenhouse gas emissions. And so we think about plant microbe interactions and what this might mean for soil carbon cycling. We found exudate metabolites and lipids might be important, but understudied component of soil carbon. So here looking at the total rate of carbon flux in exudates on the left and the amount of nitrogen here on the right, we found that the lipid fraction in exudates is about double the amount of carbon below ground compared to the hydrophilic root exudate. We also found that lipids have a markedly higher carbon to nitrogen ratio, emphasizing their importance as below ground carbon inputs. And so it's interesting to think about how this potential for carbon to nitrogen exchange between the plant and the microbes might be negotiated from these carbon-rich molecules. So in order to think about whether or not these exudates could contribute to persistent soil carbon, that's a really important part when we think about, how we could decode the rhizosphere for soil health, for example. So we want to understand this metabolic exchange between plants and microbes. And one aspect of this soil health that's especially interesting to our research team is the persistence of carbon. And so to better understand this role of lipids and metabolites in fueling microbial activity, which might result in respiration or forming soil organic matter, we calculated the nominal oxidation state of carbon. And so for our lipids and metabolites. And so basically this evaluates the stoichiometry of molecules to determine how energetically favorable the profiles are. So on the left, we have our lipids in purple and our metabolites in this peach color. For the exudates in these top two panels, and the soil in the bottom two. And generally what we see is that lipids have a more negative nominal oxidation state of carbon, which means they're less thermodynamically favorable. And so this suggests that this diversity of lipids that are exuded by roots, they can be detected in the soil, and they're not energetically favorable for microbes to decompose. So we hypothesize that potentially they could be contributing to soil organic carbon formation and persistence. Interestingly, if we compare the NOS profiles from the bare soil versus the root water wash, we find that, again, the lipids that are up here and the metabolites that are in the bottom panel, the lipids, again, they have a very low nominal oxidation state in both the bare soil and the root wash soil. And what's interesting to us is in that bare soil, we don't have new fresh inputs. So these root inputs that we're detecting in the root wash, we're also detecting in the soil. So we wonder if that's not a signature of persistent molecules that might be accumulating in the soil. So we're looking at ways to test this in future experiments. Our metabolipidomic analysis reveals a clear rhizodeposition gradient. So again, I said the experiment was designed so we could look at these plant effects starting from the bulk soil and getting closer and closer to the soil or to the root until we're in the root wash. And so that's mirrored here in our heat map. This is the median log2 normalized abundance of the top 75 metabolites and lipids that were significantly more abundant in at least one of these fractions. And what we find is compared to the bare soil here, that the root wash soil on the right, there's a striking difference in the effect of the metabolipidomic profile. And so we find that secondary metabolites, organic acids, fatty acids, these increased close to the root. Whereas if we look at the bare soil here, we found that triacylglycerols were the most represented, followed by microbial phospholipids, and very long chain fatty acids were more abundant. So suggesting that the lipids accumulating are persisting in soil, and some of these may be of microbial origin. So to better understand these signals, then we wanted to couple our metabolipidomics analysis with metagenomic and metatranscriptomic analysis. And on our metatranscriptome data, we didn't see any evidence of upregulated microbial biosynthesis of these lipids in bare soil. But granted, it's just one sampling time. But it's possible these are microbial residues or necromass that are included in the cell envelope lipids, or maybe they're preferentially accumulating due to a combination of stabilization mechanisms. And again, these thermodynamic limitations for breakdown. So we're eager to continue to test this. We did find that the rhizodeposition gradient reflected microbial phylogenetic and functional responses. So here are some highlights where we have the graphs of the median normalized expression for metagenome assembled genomes on the x-axis. So think about this as microbial taxa that have high transcriptomic abundance. And what we see is that there are certain clades that seem to be changing in abundance as we move closer and closer to the root. So these data demonstrate how roots are spatially structuring microbial communities. And while that's well known, it's interesting to see how this is associated with different functions. So below, we're looking at the community expression then of different functions, and we see that there's changes in the biosynthesis of secondary metabolites, fatty acid degradation that might be up much higher, expressed closer to the root, and finally nitrogen metabolism. And so as I mentioned in our group, our group is focused on connecting these molecular to ecosystem responses. And so these results, I think, demonstrate how we might be able to use this molecular biology of metatranscriptomes or metabolomes in order to identify, to roll them up to identify traits that might be important for example, think about bioinoculants that could help with plant growth, or microbial communities that might help with carbon storage. And so as we start to understand these associations, there's potential for breeding of plants, not only to be plant focused, but to focus on how plants and their associated rhizosphere microbial communities might be able to promote pathways that are beneficial. And we're just starting to scratch the surface here, but we think this approach has a lot of potential for identifying plant microbe interactions that may be harnessed through bioinoculants or engineering approaches. So to build on this experiment, then we did a subsequent lab experiment where we tested the impacts of drought on exudates and soil microbial communities. So this work was led by our postdoc Evan and mentored by Sneha Kovian. And what we did is we harvested mature tall wheatgrass plants from the field. These were from a site where there was no irrigation. And then we brought them back to the lab in the middle of summer and we transplanted them into pots. We gave them an acclimation period, and then we exposed half the plants to drought, where it was maintained at 5% field capacity for a period of 7 days. And then the control and droughted plants were harvested to collect both the metabolites and the lipids from the root exudates and the rhizosphere soil. And we found that drought alters root exudate metabolite and lipid composition. So in the root exudates under drought stress, we observed a higher abundance of metabolites involved in osmoprotection, antioxidation, and antimicrobial defense and stress signaling. As well, in the well-watered plants, we saw a higher abundance of metabolites involved in growth and maintenance and stable metabolic processes. And while that might not be a surprise, it's interesting to see that we can detect them and then think about what some of these most abundant molecules are that might be conferring resistance to drought or helping plant microbe interactions to be sustained under drought. And similarly, what are signals of healthy metabolism during well-watered conditions? Then we looked at rolling this up again to get at those higher ecosystem concepts. So we looked at the carbon and nitrogen content. So here we saw that drought increases carbon in those hydrophilic root exudates and in soil microbial biomass. So here looking at the total carbon in the root exudate profile or the total carbon in the microbial biomass. In the control in blue and drought in orange, we see significantly more carbon in the root exudates under drought and significantly more carbon in the microbial biomass. interestingly, we saw a significant decrease in respiration. And this, and so it suggests that maybe the exudates were contributing to increased microbial biomass, and we're seeing a decrease in respiration. So in the field, we don't necessarily see changes in respiration with our moisture treatments. But again, this might be because of in the drought experiment in the lab, we really pushed drought hard, whereas in the field, although we're limiting moisture, maybe we didn't hit the highest drought effect. So we're still interested to see from the field, we're waiting on metabolite and microbial biomass carbon data to see how similar the signatures are from the lab and field, and what these findings in the lab, how those might translate to the field. And so here, for example, when we look at these relationships, we're again thinking about how this might influence carbon use efficiency from microbes and what that might mean for carbon storage in the soil through microbial biomass and necromass, through maybe persistent dead-end metabolites, or through reduced respiration. And if we can understand what those features are that allow the plant to continue to grow while storing carbon, then we can potentially try to manage systems to enhance those beneficial outcomes. So basically, to summarize this section, we found that the short-term drought increased carbon in exudates, and that this increase in carbon was from some specific types of molecules that conferred drought resistance. We did not see a change in the lipid carbon accumulation, and there was no change in the nitrogen content of either the lipids, metabolites, or the microbial biomass. But in the soil, we did see an increase in the microbial biomass carbon to nitrogen ratio, and of both exudates and the biomass, and it increased with drought. And while we didn't see changes, the basal respiration rates of soil decreased under drought, suggesting there might be an increase in carbon use efficiency. And so there's a lot of questions that remain on how the changes in molecular composition might impact mineral-associated organic matter, for example, or persistent carbon, and how drought duration and intensity might impact molecules or carbon fluxes or carbon use efficiency. And so I think these are ripe areas for us to continue to explore as a community to understand what really drives changes in respiration versus carbon storage. And one way we've been looking at this is by trying to understand pathways where lipids and metabolites might be able to persist in the soil. So this is work done by my postdoc, Caitlin Rumford, and this is published work where this is working at a different site, but it was asking similar questions. As we think about microbial metabolism and plant rhizodeposition contributing to microbial community growth in some of these beneficial functions like EPS production, there might be microbial turnover that could lead to necromass formation and potentially persistent carbon and soil organic matter. So in order to understand that and quantify the potential contributions, Caitlin did a 13C tracing experiment to look at microbial necromass persistence. And so here she looked at, we wanted to understand how mineralogy might affect necromass retention, how annual versus perennial crops could influence carbon accumulation, and basically what forms of microbial residues form durable carbon. So in addition to necromass, if that's like the cell walls or the persistent parts of microbes, there could be other components of microbially derived organic matter that might be persisting in the soil. So she looked similar to Snea at lipids and metabolites, and using a tracer after 1 year, she found that the metabolite and lipid pools, the mass of microbially derived carbon was similar to what we found in the unextractable mineral-associated organic matter. So basically, she fed this, in an incubation, she fed 13C to the microbes. We saw it was rapidly taken up into microbial biomass, and then continued to water those incubations over a year. There were no plants in them. And at the end of that year, what we see is there's still persistent metabolite and lipids in the soil, and not a trivial amount. So we wanted to understand what specific pathways were supporting it, and we're continuing to look in our research at what specific molecules might be driving that persistence, what specific environmental conditions, and how those metabolites and lipids that are entering the soil might be processed by microbes and then contributing to this persistent organic matter. And so just a couple of concluding thoughts then, as we tie all of this together. So we started with soil respiration because this is something at the ecosystem scale that we can measure. It shows plant microbe interactions and how they might have larger scale ecosystem or landscape repercussions. And we found that soil respiration reflects seasonal variation in root biomass and also the associated exudate dynamics. So we saw this really strong seasonal trend, and we could break this apart into some of the plant and microbial components. We saw that the seasonal dynamics in microbial respiration varied with exudate chemistry using those 31 model substrates, and that there was strong evidence that the respiration potential reflects plant phenology. So when we looked at the phenology of below ground roots from the published literature, we saw similar patterns in the substrate-induced respiration or the potential respiration rates of our soil communities taken from the field. So here we're interested in continuing to pull that, moving beyond model substrates to understand how the metabolic profiles and the exudate profiles throughout the year vary with plant phenology and what this might mean for managing phytobiomes in order to increase carbon storage and decrease greenhouse gas emissions. We're excited about the metabolipidomics outcomes. We think it's a really important frontier for the broader community. We're interested in how these diverse lipids and metabolites that are exuded from mature field-grown perennial plants might be contributing to some of these larger scale outcomes. So we have one of the first explorations of what a mature field-grown perennial plant might produce in terms of the metabolipidome. And what we're finding is that these might be accumulating to carbon storage or carbon accumulation through our evidence from the nominal oxidation state and how thermodynamically favorable these compounds are, and by comparing what might be accumulating in the soil based on microbial and plant inputs. We're interested in understanding how microbial metabolism is tied to exudate chemistry, and we showed that there are some ways that this varies as we go from distance from the root, and we saw significant differences in both the structure and the function of the microbiome as we move away from the root or as we move closer to the root. And so that gives us inspiration for thinking about how those relationships might be managed to go in directions, again, that increase beneficial outcomes, providing nutrients and resilience to plants to promote plant productivity, as well as increasing persistent molecules below ground. And then finally, when we think about drought stress or changing climate, we found that in our experiments, in our lab experiments, that drought stress altered the exudate metabolite and lipid composition, where we saw an increased carbon content in root exudates, and we saw an increase in microbial biomass carbon and decreased respiration that suggests there's shifts in carbon use efficiency. And so we did not see those same respiration responses in our moisture treatments or our irrigation trials. And so we're curious to learn more about how these relationships in the lab might reflect what's happening in the field. And we all know that extent of drought can have a strong influence on respiration rates. And we were really pushing that in the lab to a drought case where in the field, I think we're a little more hesitant to stress our plants because we don't want to kill our perennial plants. But I think this is a really important area for future research of understanding how microbes can help plants to survive drought stress and what that might mean for short and long-term carbon storage. So finally, I just want to point out that some of our ongoing research, and that we're looking at this influence of plant phenology on soil ecology, and thinking both about above and below ground growth. We're interested, and so there we've got some mini rhizotron data. So we're imaging the rhizosphere over time, continuing to look at, and so that gives us insights into deeper root dynamics and what might be happening and how that might relate to some of the plant growth and above ground dynamics. We're very interested in root effects on exudate, drought effects on exudate chemistry and these plant microbe interactions. And we're just beginning to scratch the surface on how the specific metabolipidome might influence plant microbe interactions. And what I've shared with you today is thinking about the root exudates and how those might influence microbes. There's likely the other direction of how microbes are responding to this drought stress and the plant chemistry, and if there's feedbacks there that help confer resilience to the plant under drought stress. And so we're really interested in what some of those positive interactions or beneficial interactions might be. Today, I talked a lot about these community interactions, and we're specifically interested in how we could understand bacterial, fungal, viral ecology and the effects of exudates. So thinking about specific groups of organisms and how exudates from the rhizosphere might influence those interkingdom interactions. A lot of the metagenomic data, what we can annotate tends to be bacterial. We've got some fungal specific and viral experiments going on to try to integrate our understanding and have better representation of some of these other communities that are highly influential in the soil. We're always interested in translating science between the lab and the field to discover what these governing mechanisms are and what might translate from our field experiments to other contexts, other soils, other management systems. And then finally, we are working to identify what are field relevant reactions or phenotypes that alter soil carbon storage and cycling, and thinking deeply about how we couple our lab and field experiments in order to identify phenotypes that are relevant to the field, and also how to decompose or understand the phenotypes we see in the field and decode what those molecular mechanisms are so that we can start to predict outcomes and even manipulate or manage for more beneficial outcomes. And with that, I would be happy to take any questions.
Thank you very much, Kirsten, for a very informative presentation, a very intriguing work of study that I think has many applications. We do have a number of questions coming in, and so while Kirsten takes a bit of a breather and maybe a drink of water, I'll just remind our attendees that the Q&A panel is where you submit your questions. And there is a feature in that panel under each question that is the thumbs up icon. So if you would like to upvote a question to make sure that it is answered live, please click on that thumbs up icon, and it will allow us to ask those questions live. And then we'll work with Kirsten if there are questions that we're not able to get to today to make sure that we do so in a different manner via email or virtually of some type. So let's get started, Kirsten. We do have several questions. So the first one is, do drought conditions lead to increased levels of biofilm in rhizosphere bacteria?
Thank you for that question. We didn't directly test that hypothesis, and we did not see evidence for that in our data specifically. And so I think it's possible, and it's yes, there could be increased biofilm formation in order to protect microbes from that drought stress, from increased water availability. We didn't specifically see it in our data, but it certainly is an important mechanism that can help with water retention.
Okay, great, thank you. Our next question is just thanks for this interesting webinar. What is the significance of lipids in root exudates rather than organic acids?
Yeah, so the organic acids, so lipids, so they have different functions. Or from the microbial metabolism perspective, the organic acids, what we're seeing is that they are more bioavailable for the microbes. They're easier for them to respire. We tended to see them more universally used by the microbial community throughout the year, whereas many of the lipids are constrained to certain seasons. They tend to be more complex molecules that are harder to decompose. And so they're less bioavailable and they're less thermodynamically favorable. So what we anticipate and what our data are starting to support is that those lipids seem to be persisting in the soil. And we saw lower rates of respiration with the lipids. So there's a possibility that these lipids could be accumulating in the soil, contributing to soil organic matter, and potentially even sequestering carbon as plants are shunting photosynthate as exudates in the form of lipids. We find that organic acids are really more bioavailable. I forgot to point out on that slide, but I meant to, the respiration rates, so the x-axis on those slides are quite different. And what we found was that the respiration rates of some of those organic acids was really quite a bit higher than things like lipids. And so what that points to is not only the seasonality of it, but the extent of respiration. So when we looked at oxalic acid, it was about 1,500 nanograms of CO2 carbon per milligram of substrate added, whereas the lipid was more around like 20. So huge differences. I could go on. I think that's a really exciting question.
Okay. Okay. Can you explain how microbial biomass increases underwater deficit stress? And is it the fact that the stress didn't last too long, or was the stress not severe enough?
Yeah, so the stress, we use the definition of drought, which in the literature, and so we think we hit drought, and our plants were very sad, and there was a lot of panic that they might not actually recover. So I think we effectively hit drought, and that it was long enough. We're really curious why biomass increased with the water deficit. We find that to be a curious result. And it was a really surprising result for us. So we're still, like I said, this is a lot of what we're sharing was preliminary, unpublished data. And so we're investigating what might be driving that increase in microbial biomass. As we think about substrate availability, it's possible there might be some root turnover below ground that microbes are feeding on. It's really unusual though, because we would expect that decrease in moisture would decrease substrate availability and really decrease microbial biomass growth. So it's a surprising result. And we're, again, like I said, we haven't validated this in like a field experiment. We didn't necessarily see those same results in the respiration, and we're curious about what might be driving those differences. I think there is a lot of intrigue in the literature about what happens after drought. One of the really intriguing things that our group is working on is thinking about why there's a disproportionate amount of CO2 released when drought is over. And so there are some dynamics that happen during drought that are not well understood. And when ecosystems are relieved from drought, there can also be really big fluxes, disproportionate fluxes in carbon that have remained a mystery for like 50 years. And so we're working hard to understand that. I think it's an important question. And hopefully by the time the paper is published, we'll have some more insights on what might be driving those differences.
Okay, we have about a minute left, so I'm going to ask one quick question to conclude our Q&A session. And then we'll try to get the rest of the questions that we weren't able to get there. But do you focus on natural microbial communities, or is a portion of them synthetic communities for future to harness the microbiome for sustainable food production?
Yes. So
May not be a simple question, but
well, I think it's such an important question. So in our research, we're focusing on natural wild microbiomes. So what's in our native system? We have developed, if I think Ying was passing around some of the URLs. If you go to our website, we do have some reduced complexity consortia that we can manipulate that we've been working with that are, again, naturally derived. They're enriched from the field. And what we've been doing is we have been proposing some work to modify those and think about ways that those could be modified for other beneficial outcomes. So currently our program is only working on the wild, but we've got stuff in the works for some of these more synthetic biology approaches.
okay. Well, thank you, Kirsten, for an excellent discussion today. Thank you to all of our attendees, and wishing everyone a very happy and healthy holiday season. And we hope to see you all in 2025. Have a great day.
Thank you.